of the PPT. So this is the screen. So after my are you able to see the screen now? Prashant?
Yes, Pushpendra.
Okay.
Yeah.
So the screen is visible.
Yeah.
And
You can make it full screen. Yeah.
Yeah. Okay. So this was during my postdoctoral work that I was working on molecular epidemiology of leprosy, and we were investigating leprosy in humans as well as in armadillos. And at that time itself, we had this collaboration with the National Hansen's Disease Program in Louisiana State University in Baton Rouge. So, coming to a very brief background, that I did my MSc and PhD from Jalma Institute, where I was working on tuberculosis drug resistance mechanisms. After that, I went to Professor Stuart Cole's lab in Switzerland, where most of our work was on the genomics of mycobacterium leprae, and that involved primarily molecular epidemiology and phylogeography. So those are some of the terms that you would come across in if you are studying bioinformatics or any kind of epidemiological studies. So phylogeography basically relates with if some strains are related to a particular geographical region or geographical area, that basically signifies that there is a phylogeographical association between those strains and that particular area. So our main question that we were investigating was about leprosy in humans. And most of us, you would think leprosy as a disease of past. But this is not the case. Nearly 1/4 new cases are recorded globally. Of them, 60% are in India, which is very unfortunate. We are only 18% of the global population, but we have disproportionately high burden of leprosy, which is around 60%. This can cause deformities due to loss of sensation, and bacteria remains uncultivable, and we do not know much about its transmission. We know that it has very low genetic diversity, which makes it very difficult to perform any molecular epidemiology using PCR. That is why we need some more detailed investigation in case of leprosy or other bacteria which have low genetic diversity or which are difficult to culture. So I'm coming to the case study 1 about leprosy. We know that has very little genetic diversity, and we don't know how to confirm whether two strains are same or not, whether two humans are infected by the same strain or they have different source of those strains. This slide here says that despite large decline in leprosy, the new case detection rate shown in this dotted line has not come down significantly over the years, and the same situation still continues. In another 10 years also, that has not significantly come down. In fact, during the COVID-related lockdown, this situation has become worse because now more number of new cases are being detected. So the only animal model for leprosy is armadillo. And you can see this picture. In this picture, you cannot probably guess the size, but it is a cat-sized animal. You can see here, this kid is holding the armadillo pup in America. So these armadillos are actually very popular animals in southern United States. People make a lot of different type of designer bags. You can buy them from the Amazon. But I think after hearing this presentation, till I finish the presentation, do not yet order anything, please. And we will see why. So this was very well known for 20 years that armadillos carry leprosy. And even before they were ever used in the leprosy research, it was known that they carry natural infection of leprosy. But people still used to keep on interacting with these animals in many ways, either as a roadkill. So you can see that this armadillo is trying to cross the road, and if you cross from Texas to Baton Rouge, it's around 200-mile distance, and you would see at least 5 or 6 animals uh, which are crushed by a car or some other vehicle. But still, uh, these are considered harmless animals, and a lot of people like to eat their meat. This is considered as a delicacy, and the way this man is cooking this armadillo for barbecue is actually is not sufficient to kill any organism which is inside this flesh of this animal, because it would not kill the entire bacteria which are inside. But when we studied these animals, we were investigating leprosy in animals, leprosy in humans from United States, and we were recording if they have been to any leprosy endemic country or whether they had immigrated from, because molecular epidemiology will basically involve uh, epidemiology first. You need to, if you just have sequence and no other metadata or other information, it will not basically be very useful to derive a very strong conclusion. You can derive some inferences, but to derive some conclusion, you need supporting evidences. That's why it's called molecular epidemiology. In addition to molecular biology and bioinformatics, it requires metadata which comes from epidemiology, such as what was the history of that patient, whether there was an index case of leprosy in the family or in the neighborhood or in the workplace, or has that person been to a leprosy endemic country in last few years. And in leprosy, the incubation period is up to 5 to 7 years, and this can be even up to 30 years in some cases. So that is why it becomes very difficult to derive uh, any conclusion immediately, because disease may manifest after several years of exposure or infection. So this is the figure which was published in the New England Journal of Medicine that Prashant was referring, and it was in 2011 that this study was completed. You can see that if you don't see, let's think of some simple this figure in a simple format. It's like grapes, those small yellow. If you can follow my cursor, are you able to see the cursor on your page?
Yes, sir.
Okay, so you can see that those are small dots, small circles of different colors, which basically defines what genotype they belong to. For example, yellow color is of genotype 1A. So the samples on this side are of a particular genotype of 1A. We'll see how this is derived later on. But let's first see what basically bioinformatics can tell us about some findings once we have the data. Once we know that this is the genotype, this is the strain, and we need to have, and then you are seeing some asterisk mark here, for example, which means that this person has been to some leprosy endemic country. So it has some foreign endemic exposure. Leprosy is not endemic in United States. Most of the cases are from the immigration. So you can see that there are a lot of these asterisk mark in different colors of those small circles, and then we are coming down towards a bigger circle, but till there also we have a lot of these asterisk mark, meaning that they have foreign endemic exposure. But here there is a big circle. It seems like we are comparing kind of grapes versus some big fruit like pumpkin. So here one small circle means one case, Whereas here you in this where you are seeing my cursor, it has 28 armadillo derived leprosy strains and 25 from humans. So nearly 50, so these 53 cases are exactly identical at the genome level. We are not talking about just PCR based methods. So at the genome level, they have almost identical DNA. And this is very, very surprising in field of leprosy. When you have this type of, because leprosy strains are uh, not clustering so much, at least in countries like United States where there are not so many cases, and if there are some cases, they are from different countries. That is why they are not clustering all over here. But in this, we have a large number of samples clustering with each other, and then there are few which are kind of emerging from this big cluster. And the small numbers that are written, for example, 2 here, 3, 29, those are the differences in terms of the SNPs between these two strains. So there are 34 differences between this and just two, whereas on this line there is just one difference. So this is not a very complex figure. Basically, I have probably skipped some of the background because our main aim is not just a simple scientific talk, but to have some hands-on session also later on. That is why I am trying to skip some of the background. But this is far more simpler figure than a lot of molecular epidemiological phylogenetic tree that you see. But this paper was entirely based on this figure and got accepted in New England Journal of Medicine because the evidence of having identical strains of 28 armadillos and 25 human, and most of those human samples had no history of travel to any leprosy endemic country. So they acquired their disease from some local source. And what that local source can be, that local source, from this figure it appears that it can only be armadillo because they are having the same identical strains. Still the journal asked us to use the word probable zoonotic leprosy in this publication. Probable zoonotic leprosy in the southern United States. We were okay even to write a probable word because in case of leprosy, it's very challenging to show the direction of transmission from this person to another person, this spread. This is difficult to prove in case of leprosy. But how we came to this, what was the type of data that we used for this? Not all the samples were sequenced for its whole genome because, as I said, leprosy bacteria, which is called Mycobacterium leprae, is not a culturable organism. It is always with some clinical sample. So you would have to basically deal with large amount of human DNA. If you sequence it directly, more than 99.9% of your sequencing reads will align to human DNA. Because human DNA is 1,000 times bigger and 1,000 times more abundant in a human tissue. Or in armadillo tissue, the armadillo DNA will be dominant. So that is why we had to rely on some PCR based studies also for most of the samples. But for few samples, we were able to sequence the entire genome also. But what type of data that, how we compare those PCR based sequencing, we will see in the hands-on session today. But let's see another molecular epidemiology study. So we knew that these armadillos and human have the same strain. And what we knew at that time was that leprosy was not present in America before Columbus discovered America. Because all the skeleton sample of described from USA, if they are 1600 before the 16th century, they were generally found to be free of leprosy. So we know that leprosy strains reached America in around 16th century through colonization. And we know that armadillos are not found outside of the America or the New World. In North and South America only, armadillos are found. That means armadillo got leprosy from some human contact initially, and then now they are amplifying it. And when humans are interacting with these animals, either as a food source or by some accident, or because these animals are present in the gardens, so there is some exposure to human from the bacteria present in armadillos, and that is probably giving the humans this disease leprosy. So initially it was from human to armadillos, but then in armadillo they have multiplied in large numbers in armadillos, and when they come in the garden or when somebody eats it, then there is a high chance of those bacteria entering into human. And if that person is genetically susceptible to leprosy, in few years they may develop some signs of leprosy, but not all humans are susceptible to leprosy. So that is another component of molecular epidemiology. We are investigating an infectious disease through molecular epidemiology. So we study bacteria, its DNA, we study the environment, we record their demographic details such as age, gender, their occupation, their age, and then what type of exposure they have from index case, from family. Or so index case is someone who was identified with that disease before other members. So that is generally in a family, the first case which is recorded is considered as an index case, and then when we see other members as a follow-up, the other cases are considered as the contacts. So same was the case with the corona also, but corona was having much higher infectivity with a very smaller incubation period. So let's come to the another story. We are here to learn about the application of molecular epidemiology, and then we will learn about what are the tools to analyze the DNA sequencing data. But let's first see the stories and the case studies. So we know that the leprosy reached from Europe to America in around 16th century through colonization. But leprosy had declined in Europe after 14th century. Until 12th century, there were a lot of cases of leprosy in Europe, which is evident from lot of records, historical records as well as archaeological samples, such as these skeleton sample from 10th century. These have characteristic osteological lesions, because leprosy can cause loss of sensation, and that leads to improper distribution of weight, that leads to fusion of some bones, and sometimes secondary infections due to loss of sensation also the secondary infection also cause this type of deformities that you are able to see in the pictures. These digits of hands or foot are lost because of the secondary infection. But this is very much traceable in the skeletal samples also, even after thousands of years. For example, all these are shown here, and the nasal septa is actually damaged by a large number of bacterial multiplication in the nasal mucosa area, and that leads to this sinking nose picture, or this is called the face of leprosy, because this nose bone is actually damaged. So this leads to this type of characteristic picture. So after the whole genome sequencing of, we are not covering more details, but this is mainly a hybridization-based capture of the target DNA. Using that hybridization-based capture, we were able to sequence the whole genome of these M. leprae strains from 1,000-year-old skeleton, which are shown in the red font here in this phylogenetic tree, and this is based on the whole genome sequence data. So in this phylogenetic tree, you are able to see that this Jorgen 625 sample from Denmark, which is nearly 1,000-year-old, is coming very close to this cluster of NHDP63, which is present in armadillos. This is called V1 strain. And the same strain is also present in one of the skeleton sample. It's called SK2. SK means skeleton from the United Kingdom. So this 1,000-year-old skeleton sample, exactly this one actually, this skeleton sample has nearly same identical genome, only 21 differences on this branch, here, and 7 differences on this branch, in this branch number 3. Both are the same genotype, and at the genome level they have lot of identical in many molecular markers. So we consider them as the identical strains. In even 1,000-year-old sample, we are able to cluster them together. In so many years, it has not actually changed much. And this also tells that leprosy declined in Europe, not because of the loss of virulence, but maybe some other factors such as the host genetics might have played some important role, or when 14th century plague had killed half of the European population, that might have changed some social structure. The population density came down, and those changes might have contributed mainly towards the disappearance of leprosy gradually. And after 14th century, there were very few cases left. But up to 10th century in Europe, there were more than 20,000 hospitals for leprosy. Those were mainly the isolation centers for leprosy patients. So basically, this is another molecular epidemiology of, or we can say, ancient zoonosis. There was recently a paper where they confirmed this for squirrels also in Europe. European squirrels, red squirrels particularly, were having exactly identical strains which were present in the skeleton sample. So that is another ancient molecular epidemiological investigation, or this is also an example of ancient zoonosis. So most of you are familiar with word zoonosis, that when a disease comes from animals to humans, that's we use that term. So this was published in Science Journal in 2013, 2 years after the armadillo story. So this was also very widely covered. So I would basically stop here for the hands-on exercise so that we can utilize the sequence visualization tools to know how to come to these type of conclusions in terms of molecular epidemiology when we have Sanger sequencing data available with us. So I'm asking Afjal to come here and please open those code-on-code sequences. I hope most of you have downloaded the code-on-code software on your computer.
Yes, sir.
Okay. So please open code-on-code on your computer, and in form of new project, you would so in the code-on-code software, you are able to see open new project or after opening the computer. Initially, it gives 1 month free trial, and then so let me share the screen again. So are you able to see the screen now? So there are multiple windows, but you are able to see the quote-unquote aligner window now? Yeah, I see that it's visible. Uh, in the unassembled sample, so basically when you have this window open, you can either say new project, or and in the new project you have this type of folder called unassembled sample without any sequences inside it, and then in this folder you can, so let me just ensure that you are on the same window, or first we can demonstrate it and then you can start testing it with some of the uh, test samples. So I would prefer that let's begin with the functionality of different uh, functionality of this software, which is for the Sanger sequencing data analysis primarily. So in the file, you would have this option of starting as a new project, and once you have opened it, there is a possibility of importing samples. You can add samples. You can import your sequencing data either in form of a folder, which is or you can import sample one by one also. So when you click add samples, you would see this window, and in that you can browse it from wherever you have stored your sequences. So in this folder you can now see that there are several .av1 files. Those are the applied biosystem Sanger sequencing files. So you can click all of them. You can select all of them and import them, all of them together. So now they are, well, if those are already present in the your unassembled samples, then it would say that would you want to rename them, or you can say ignore duplicates also, so that you don't want to import the same sequence multiple times. So after that, you have these 105 files here, and those are given their names. We don't know which gene it is, whether it is from the forward primer or from the reverse primer. But let's see how the data looks like from the Sanger sequencing. So I would encourage you to unmute your microphones, and in case you have uh, any doubt, you can immediately ask. So how many of you have worked with the Sanger sequencing data? Anyone who has worked with Sanger sequencing?
Yes, sir. We just use it to sequence the 16s of bacterial samples. So we just assembled the forward and the reverse read of single bacteria.
Okay, good. Hemani, so you have used it. And anyone else? I think
Yeah,
most.
I have also used Sanger sequencing, but
And
but
what
that's for
you are using for the alignment, uh, for for the assembly and comparison?
I was using Chromas Pro software.
Okay, so there are multiple, like such as BioEdit, Sequencer, and maybe several other tools, also DNA Star. But
Yeah, DNS star also. I have
yeah,
acquaintance to DNS star, but basically I was using Chromas Pro, more like for thalassemia samples and other variant validations, no, from whole exome, clinical exome sequencing and all that. So we were
okay.
doing it.
So basically, you are familiar with overall chromatograms and such things. I think for other people also, I would just basically, whoever has not used this before, I'm highlighting few points which are important when you want to derive any conclusion, not only molecular epidemiology, but if you are studying drug resistance, whether you are determining genotype, or whether you are identifying the species based on the 16S or anything, you have to basically get rid of the low quality reads. There is no choice, uh, or there is no point in having sequences which are actually of very low quality. For example, this particular base is referred as G, and that G has a quality value which is indicated on the right corner of this window. It has only quality value of 5. And then the next one is an A. But you can see that there is a chromatogram of red color which signifies a T, and then a green is for A, and blue is for C, and a black line for G. So all 4 bases are there, and its quality is even less. Quality value is only 4 here. So all such sequences need to be removed. And this software basically gives some easy visualization by putting them into green highlight. So green highlight doesn't really mean that this is good data, but it's opposite here. When it is highlighted, that means it's actually indicating lower quality of the data. So you can simply select the sequences that you think are of lower quality, and then you can press the backspace or the delete button, and that would basically backspace button will delete all such sequences of low quality. Similarly, on the right side, there are some low quality sequences. You can delete them also, and now so this will take a lot of time for doing it for 100 samples. But there is a simple tool which is indicated by a symbol of seizure here, uh, which is basically indicating clip ends, or you can see it from the in the sample. There is a option called clip end, or there is a shortcut command also, Control Alt E, which is written. You don't have to remember them. You can just select the entire folder and press this button, clip ends. So that will clip the ends, and it also indicates what is the uh, how many samples will go to the trash, meaning they have very low quality. So we can see directly that these 3 samples are already sent by the software in trash, and we can see that what type of quality they have. It's actually not very meaningful data. If you use this for any 16S based or drug resistance based studies, those would derive a lot of errors, and those conclusions are not actually uh, reliable. This also, and in all such sequences, if there is a non-specific amplification or the primer is not optimal, or if wrong primer was added, all those errors can actually be causing this type of failures of sequencing. So yeah, we can see that out of 105 samples, 3 are already sent to trash, and we need not to waste time on analyzing those. We simply may need to resequence those or have to re-amplify them by. And then if you press on the in the So in this option, there are multiple options on this software. File, edit, go. Go is for the blast search. We'll come to that also later on. And in the sample, there is a clip and functionality we just saw. And then in the config, there is an option called assemble. So once we click that assemble, it has already assembled those samples into config 1, config 2, config 3. In the config 1, there are 48 samples. So without actually assigning any name of the gene or any forward or reverse primer, we already see that all 48 samples are in one config, and when we click on the config, we can see their alignment with each other. If there was a reference sequence to import, that can also be imported, and there is actually a reference sequence already embedded in this. If not, we can import a reference sequence simply as a .txt file. That has to be a text file. When you click on the text file, there would be no chromatogram because this is only a text file, but this can also be aligned. So here you can see that the text file of the sequences that were imported, and this is a reference sequence of the target gene in mycobacterial DNA. That codes for RPOB gene, which is the target of rifampicin drug. If there is a mutation in RPOB gene in particular areas, that leads to the drug resistance. And drug resistance is also an important molecular epidemiological indication. For example, if you have 3 strains which have same mutation, and that leads to drug resistance, and all other samples are actually wild type, then you can think that most likely the ones which are resistant were probably through some common source, possibly. It's not always the case, but if you want to investigate them, those 3 would be important, because if they are having same mutation which is causing resistance also. But in this alignment, you can see again there are some in the red font here. That means from the those bases are different. Let's see whether those are really the same base or not. So you can now see that when it says A, there is also another base at the same place, which is of the black color, and that indicates a G. So this sample has 2 peaks. One is of green color. Green color is for A, and the G is of black color. So now 2 samples are shown when we click the same area. So in this upper sample, which is 001 sample, there is a clearly single peak of only one base, which is a G. But here we have both the bases, G and A. Unless we have a reverse primer for this same sample, we cannot conclude that this is a mutation. Sometimes for the heterozygous bases, that is a different case, but today we are studying molecular epidemiology of microbes, which are not diploid genomes, but there is only one chromosome in most of the microbes. So having this type of basis is only possible when there are mixed population or low quality of sequencing data. If there is a mixed population, then you would see it in multiple places. But this here usually mentions that there is a low quality data, and we have to be careful about deriving any conclusion about molecular epidemiology or drug resistance from such type of chromatograms. Likewise, so once you have basically manually checked some of those sequences, for example, if there is a mutation in some of any sample in the consensus sequence or in the contig, the last line indicates the base which is most common among all the samples. So here A is indicated, meaning in most of the sample here it's A, but in some areas there is a different base. For example, it's indicated in red font here in sample number 43. Here there is a C, and we can see whether the quality of this base is correct or not. And again you will notice that there is a double P. This C could be an A also, and in most of the sample it is an A. So unless we have a forward and reverse sequence for this base, we would not be able to conclude that this is actually a mutation leading to drug resistance, or this is a particular genotype of this bacteria or virus. So these are few examples from this alignment. Let's come to another contig. If this contig has many samples, there is another contig which is of 22 samples, contig number 3. You can rename this contig by clicking on this contig and here. So now you can name it as RPOB because you know that it has RPOB and how many. So RPOB sequences are there. And let's see the contig number 3, which has 22 samples. And here also we have a reference sequence, which is MLEPRI-FOLP drug resistance determining region. So there is only this small area of sequence. And we know that in this particular area, if there are any mutations, then this leads to resistance to ofloxacin. And you can see that in this entire area in the contig 3, the sequence that I highlighted, and probably you are able to see the cursor here. So there is no red color letter. So anyone can tell from the previous discussion, just in the previous example, what does it mean? In all those 22 samples, the sequences are identical. All of them are identical to each other because there is no red color letter in this area. For example, there is a T here in the red color because there is a C in one of the sample. But in this particular area, which is in the consensus also, in this particular area which is highlighted, there is no change in the sequences. And the reference sequences is from a wild type bacteria, which is not resistant to this ofloxacin drug. So what type of conclusion we can derive from this, that they are having the same sequence as the wild type? What does it mean? Whether the samples would be drug resistant or sensitive? Hemani or Pranit, anyone? Angu?
Resistant, sir.
What would it be, resistant or sensitive? So let me explain again. This reference sequence that I have selected is from a wild type strain. Wild type means it is not resistant. Wild type means it's a sensitive strain.
Yes, sir.
And
Then
the
it will be sensitive.
And then all
Then
the sequences, all the sequences are same as the wild type sensitive strain, meaning all the samples are sensitive. So in just maybe 2 minutes or 3 minutes, we were able to conclude that all these 22 samples are wild type, having no mutation. Likewise, if there was a particular mutation which is indicative of of a particular genotype, let's say genotype 3I, then you would see that if that mutation is present, then all the sample, whichever sample has that particular mutation, it would have that particular genotype. If two genotypes are different, we can rule out that they have the same source of infection because they are of different genotypes. But if they are same genotype, then we have to look into more details whether they can have further some other differences in terms of their drug resistance profile, or they have some other mutation in a different gene. So those would basically be more detailed molecular investigation required. But if there are two different genotypes, then you can directly say that they are not from the same source. So they might have acquired it from different sources. So that can be a molecular epidemiological inference from having different genotype. Similarly, coming to the contig number 1, here also we have a reference sequence indicated in the small letters. And this reference area is highlighted here from the reference sequence. And you can see that in one of the samples there is an insertion of an A, but in all the samples there is no base corresponding to this particular area of A. When we double click this A, there is no peak actually which corresponds to this sequence. A is such a small signal here, well below the background, and its quality is also very low. Quality is just 8. So actually there is no such base. So we can simply delete this A, and now this deletion would be gone from the contig. So for any type of molecular epidemiological investigation, it is very important to really retain good quality sequences and remove all those which are not reliable signals. For example, so from this conversation, we are only first ensuring that we have good quality sequences. For example, in one of the sample here, there is I have highlighted called C. It's in position 135, and when we click on this base, there is a C which is of very clean quality. There is no background. Let's zoom more into this. Control and but are you able to see that the base that we are seeing here?
Yes, sir.
Yeah, it's a C, and there is no ambiguity. There are no other backgrounds. Whereas in this sample, there is a T. So now you can see that one sample is clearly having T and other is having C. So there is really a mutation. In reference sequence, there is a C. Meaning there is a, now we have a reliable example of a mutation. But does that mutation do any effect on the protein? Because now we have the entire reference sequence of this particular gene called GYRE. This is the DNA gyrase gene, which helps in unwinding of the DNA during DNA replication. So it is starting from ATG, meaning that is the start codon. So let's convert the sequence into protein sequence. this is the DNA sequence. So let's convert it into protein sequence. And that can be done from this cursor. It's switch between bases and amino acid for sequence. If you are able to follow the cursor, GAT to ASP, when we click that, it has converted the DNA sequences into protein amino acid sequence. But we know that it is not starting in the right frame. Hemani?
Yes, sir.
So is this visible on the screen? If
Yes,
those
sir,
who
it's
are
visible. Sir, we
okay.
can zoom our screen to increase the
Okay,
visibility.
good. Those who are browsing it on their mobiles may have difficulty, but so now we know that this is not a correct frame because this is not starting with ATG. This is a complete gene. So what should be the methionine? First
Methionine,
ATG. Yeah,
ATG.
methionine codes for ATG. Methionine is coded by ATG. So because this was not the right frame, so you would click it again on the same. So now it will select another. So now it's back in the DNA sequence, but clicking it again will put it into protein sequence, and now you can click on the next frame. So now this has converted into methionine. So that is the right frame. It's now starting with methionine, valine, serine, aspargine, like that. And if we keep on going through that, it would indicate that the first mutation that comes is histidine in the reference sequence. So in the reference sequence, let me bring it. So histidine has, so this histidine is converted into tyrosine. This histidine is converted into tyrosine. In most of the sample, you are able to see histidine, but in some sample, including, so in the reference sequence also it is histidine, but it's converted into tyrosine in some samples. Yes, in 3 samples. This one is tyrosine, this one is tyrosine, and there is one more. Yes, this one. 2 are together here. So that is that a synonymous mutation or non-synonymous mutation? Angu or Saima?
Non-synonymous, sir.
Yes, because amino acid has
Non-synonymous.
yes, it's a non-synonymous mutation. Then there are several tools which help you identify what is the effect of that non-synonymous mutation. Whether there is a major impact on the protein structure or whether there is a very slight effect, or sometimes there might be some polar amino acid to non-polar amino acid also. So those changes would be difficult for protein functions. If those that particular motif of protein is important for a particular function or particular conformation, then it may change the function or the stability of protein. And if that particular site is related to the drug binding site, in that case, it can even lead to drug resistance. So those are some examples that we wanted to study. And in case this particular area is histidine in a particular genotype, then also you can say that this is a genotype C, a particular genotype 3I, whereas the other sample could be a different genotype. So those are few inferences from the Sanger sequencing data, but it would not be based on just one sequence. You have to basically, you can rule out if they are not the same strain by a single evidence also. because you have found an evidence that says that they are different strains. But if they are same strain based on one sequence, you still have to analyze few more sequences to prove that they are identical in those areas also. If that sequence was not the correct one, you can, it could be actually reverse complement also that you might have imported. So in the, let's see if you have this, I'm converting it back into the DNA sequence by this option of GAT to ASP here, because I'm more comfortable seeing the DNA sequences than the protein sequences. For some reason, I have some fear of proteomics. Even if I see the codon instead of nucleotides, it makes me feel uncomfortable most of the time. So now we are back into the DNA. It's much more simpler than the amino acids. And let's see which, because I mentioned you that this is a gyre gene, but if you don't know what sequence it is, then you can select particular sequence and you can select the good part of that sequence only. Let's say this is very clean sequence up to here. You can select this area and you can click the sample in the go option. So we want to go to blast search for this particular sample. So you select the blast search in the go, in this menu go, and then in the blast search you can select nucleotide blast or the blast N. There is a shortcut Control Shift B, and based on that, only for the area that you have selected, it would put it. It will bring you to the window where you can perform the blast search without having to copy paste or name the sequence. And in the nucleotide blast, let's again do that in the go to blast search, nucleotide blast. Probably it's requiring a login. But once you are logged into your NCBI account, or you can also copy paste the sequence, but then you have to also give the name.
But earlier we never used to never required to log in
Yes,
for blast search.
I
Is
also
it,
remember that actually, but it
Hannah?
seems to be
Is
a
it some
new.
new?
It may seem to be a new feature, but yeah, earlier it never required login. Uh, Maybe there is some conflict in some login in this, but it will automatically take you to the blast search and will help you confirm that the sequence that you want to align is actually of that particular species or not. अगर वो मेरा वाला login से करके देखो ये रहा तो earlier it never required any login actually for me also. If we select entire sequence, so let's copy and paste that sequence in the codon in the blast search. So nucleotide blast, we have to provide the form that's usually better. Ref gyre, we know that what sequence we are pasting, and now just simple blast search highly similar sequences. or the somewhat similar sequences, blast time. And then on blast, it will indicate what sequence. So basically I explained some of the quality control measures for performing any molecular epidemiological investigation if the Sanger sequences are being used. So it's indicating alignment with the uncultured microbial species clone GTML, and it's indicating gyre gene, partial uncultured microbial species. Actually, this is a microbiome Leprosy sequence, but somebody has submitted this as a uncultured mycobacterial species clone. Here you can see that Leprosy gyre subunit gene. It's indicating 100% identity with these sequences. You have no gaps. All 141 bases which are 100% identical with the Mycobacterium leprae gyre subunit gene. And those who submitted these sequences probably didn't match it with M. leprae first. Otherwise, they wouldn't be mentioning it as a mycobacterial species, but they could have named it as a Mycobacterium leprae. However, in some conserved genes, it's difficult to conclude from just one gene because that area might be a conserved part of the DNA rather than really that particular species only. It could be some other species also which is having same conservation. So basically from me, this is all that I wanted to tell you. Once you have those good quality sequences, there are several other tools for deriving further molecular epidemiological inferences about whether they are, how close they are in terms of phylogeny, or how long they have diverged from each other. If you know other details such as the year of isolation of the samples, and based on that, mutation rate can be estimated. Sometimes the evolutionary dynamics can also be estimated if you have samples from diverse time frame. For example, the 1,000-year-old skeleton sample helped us in determining the most recent common ancestor of leprosy strains. So all those are actually part of molecular epidemiological investigations using different tools, and we are not going into those for today. So I'm open for questions from all the participants, and I'm sorry for the technical glitch in the beginning. I hope this was useful to some of you, and at least you are familiar with the role of quality control measures for sequences rather than, because I have seen a lot of people directly using BLAST tool for the text sequences from the chromatogram files. They, without doing any trimming or without doing any confirmation, they directly report that this is a mutation present here, or this is a new species not aligning with any known. That mostly is because of not really trimming out low quality areas of the sequence. So yeah, I'm open for any questions from all of you, whoever has. We would learn together. Anuraj or Simon,
Good evening, sir.
good evening.
Sir, I'm undergraduate third year student, and this is new to me. So, for a beginner, how shall I proceed?
Okay, so you have downloaded the sequence, the quote-unquote aligner software.
Yes, sir.
So that would be freely available. That would be available with full functionality for 1 month.
Okay, sir.
And there are some example files in the project. When you download it, there are some example files also. And if you send your email, we can send you a few chromatogram files for practice, along with the reference sequence that just now I have shared this gyre sequence. with the reference sequence. Afzal will send to you. You can send your email through this WhatsApp group. We'll share the example files for your practice. Let's initially begin with the drug resistance, and for the molecular epidemiology also the same approaches will be useful.
Okay,
It's
sir.
mainly for identifying polymorphism, whether that causes drug resistance or that is linked to a particular genotype. That's all a different thing, but the process is same. We want to identify the changes in DNA, those polymorphisms, whether there are some insertion, deletions, those also sometimes are useful. Yeah, I hope I answered to some extent.
Yes, sir. Thank you, sir.
Or anyone else? I will see, sir. So Abdul has stopped sharing now, and those who have not used this type of sequence alignment software, can they raise their hands on their windows? Sujit, Pragati, and okay. So for all of you, we would send some chromatogram files for practice along with their reference sequences. And in case you have any doubt, you can You can set up one Zoom session where you can practice it with one of our team members or with me until you become familiar or comfortable with the use of that particular software. So, anything else for to discuss for today? Pragati Gupta, you can also send your email on that WhatsApp group, and we can share you the raw data.
Sir, it will be great if you share raw data in the WhatsApp group that everyone can access and they can access or try this software.
Okay. Okay, then we would provide the sequences. We would put in a Google folder, and we will provide the
Drive link.
drive link to that.
Yeah, Pushpendra,
Okay.
sorry to interrupt you. I
Yes.
think, we are going to have another talk at 7 o'clock.
Okay, so we are done.
Thank you.
We are almost done. Thank you very much. Then, bye bye to all
Sir,
of you.
group pic is left.
Group picture.
Yeah, one group
Okay.
picture. Yeah. So I can be a good photographer. I already have taken earlier. Yeah. Yeah. Good thing is there is one. Neha's autopilot was there, so I had to remove. Yeah. Yeah. Thank you so much, Pushpendra, for a very interesting talk. I'm sorry I missed most of it. Yeah.
No, thank you very much for giving this opportunity to interact with all the.
Yeah. No, at all. Yeah. Yeah. So Gautam, please switch on your camera. Pragati, you have some question. You raised your hand. Please switch on your camera. Afzal.
Actually, he's in the same frame as me.
Oh, okay. Yeah. Agu Peter, Gargi, Anguraj, Yasin. Okay. Yeah. Say cheese. Cheese. Yes. Yeah. Thank you. Thank you very much, everyone. Please give a big thumping round of
Thank
applause
you,
to
Dr.
Pushpendra.
Pushpendra.
Please.
Thank you very much.
Thank you.
Dr. Pushpendra, I just wanted to ask you: do you also have deal with the human genome sequencing data as well, like Sanger sequences?
Not me, but other people like Afjal has analyzed human genome sequencing data. Arup in our team has also done analysis on the whole genome sequences of human. For example,
Like when we do variant curation for like targeted sequences or something, when we are doing at that point of time, we are not actually going ahead with all the entire gene or something. We already know the targeted region, so that kind of validation part is also been done over there, or what, like, can be done?
we can discuss that, and I think we have done this type of work for bacterial genome, and that also was done after the targeted capture of the bacterial DNA sequences. In the same way, if you are particularly interested in few part of the genome only, or in targeted sequences, that can be aligned directly.
Directly to the human genome, right? If you put up
Yes.
the human reference, like human genome, you're going to select the reference genome, the type of samples you are using, I guess. So
Yes.
will you be selecting the reference genome likewise? Na, see, you have a reference genome at the base of the window, you are at the software where you were aligning the reads from the bacterial genome, right? There is a reference for that particular species over there
Yes. Yes.
at the bottom. So when we are dealing with the human genome, so I guess we will be picking up the human genome. That does the assembly also matters there? Like we have AG37, AG38, so we have to be careful enough to pick up the assembly as well. What kind of assembly, on what assembly we are actually working, where the targeted sequence has been there.
So I think if it is a subset of the total sequence, you can for the analysis part, to make it more efficient, you need not to import all the reference genome. You can directly use the target sequences for alignment that you are interested in aligning.
Okay.
If you are
Okay.
Hello.
Arup can explain more, I think. Arup?
Hello. I would like to add some comments regarding the genome version.
Uh-huh.
So, the panels that has been designed for screening like cancer or genetic disorder,
Uh-huh.
they are designed based on a specific genome version in mind, like AG19 or AG37 or AG38.
37,
So
AG38.
definitely you first because the regions will change based on the genome assembly version.
Yeah.
So either you have to, like, if you want to, you have designed something based on AG37 and now you want to use AG38 as reference genome, then you have to do something some liftover for the target panel. Because when you are doing variant call, you are not going to do the variant call for the whole genome. It will take a lot of time and resources, and it will yield nothing because you already know the variant should belong to the target region only, rather than like flanking regions or other UTR regions or non-specific regions.
Yeah.
So in that case, if you want to use a targeted panel, stick to the reference genome against which the panel has been designed, or do a liftover for the genomic regions if you want to use a different genome version or assembly version.
Okay. Okay. That's wonderful. Thank you. Thank you, Arup.
Sure.
And thank you, Dr. Pushpendra. It was really wonderful session. This is something new which I have not used this particular software. It's going to help me a lot. Thank you so very much. Brilliant.
I'm glad to hear that. Thank you very
Added
much.
so much to the knowledge, upscaling, something really great. Thank you so very much.
Thank you. Thank you.